Here's the workflow for the Ovation Target Enrichment System, our simple single-tube solution for targeted resequencing. Input for the system is 10 to 500 nanograms of fragmented DNA, which then goes into an end repair step, followed by ligation of the forward adapters and a bead cleanup step. Probe binding and extension can be completed in 3 hours. However, best results are achieved with an overnight hybridization step of 12 hours, which in practice means the protocol will be split over 2 days. Following another bead purification is an optional qPCR quantitation step to determine the optimal number of PCR cycles to use during library amplification. After a final bead purification, the target-enriched library is ready for sequencing. The Ovation Target Enrichment System features NuGen's proprietary single-primer enrichment technology, or SPET. We start with human genomic DNA. Within the genomic DNA are regions that have been defined as the targets we want to enrich for sequencing. Generally, these are exons of targeted genes, but can be essentially any regions of interest in the genome. Probe landing zones are the areas in which the enrichment probes are designed, and these are located a short distance 3' of the target region on each strand, enabling independent coverage of the target region from both strands. The DNA must first be fragmented to an average size of 500 base pairs by sonication. The majority of fragments will not contain a target region. Here we are showing fragments containing target regions where a break has occurred within the target region itself. After end repair, indexed forward adapter sequences for Illumina sequencing are ligated to both ends of the randomly fragmented DNA molecules. At this point, each sample is uniquely tagged with an index, allowing for multiplex pooling at the next step. The adapter ligated DNA is mixed with the specific targeting probes and denatured to allow annealing of the probes just 3' of the target regions independently on both strands. In addition to target-specific sequence, the targeting probes have a portion of the Illumina reverse adapter attached to their 5' ends. The hybridized probes are extended by the action of polymerase through the target region on both strands, continuing to the end of the forward adapter, resulting in library templates containing the target region and both the forward and reverse adapter sequences. The location of the reverse adapter sequence in relation to the target region is defined by the location of the complementary sequence within the probe landing zone, while the location of the forward adapters in relation to the target region will be random, determined by where fragmentation occurred. Following denaturation, PCR primers are annealed, completing the full forward and reverse adapters, and libraries are amplified by PCR, creating a target-enriched library ready for sequencing.